So my name is Rhandzu Ndzhukule. I work at the Desmond Tutu Health Foundation. So in keeping with today, the aim of today's meeting, I'll be sharing with you the work that the foundation has done with a particular focus on adolescents. So I work at the Masiphumelele site, and not only do we have the research clinic where we do all the research work, we also have an adolescent clinic which has been operational since 2011, 2012. We provide sexual and reproductive health services to the youth of Masiphumelele. We are staffed with an adolescent-friendly team. We see roughly 3,000 young people in a year. Not only that, we also have a gym at the site where we aim to improve the health and the well-being of the young people that come through our doors, providing a threefold intervention. We provide health screening services, fitness training, and health education and promotion. So in my talk today, the 15 minutes that I have, I will try to speak quickly about who the adolescents are, why we include them in the work that we do. And it was difficult to choose only 3 studies that I will talk about, but I wanted to speak at least about the 3 studies that enrolled adolescents in this site. So I chose the Plus Pills site, and mainly because it was the first oral PrEP study in an adolescent population in Africa. I'm also going to speak about the Zoli study, the Zoliflozin study, which was the first treatment study in South Africa to have been granted a waiver for parental consent for older adolescents. And I will speak briefly about the Purpose 1 study, and I'm pretty sure that you're going to be hearing a little bit more in other talks on the study. But what's interesting about the Purpose 1 study from the adolescent perspective is that it was a PrEP study that enrolled adolescents right from the beginning. So who are the adolescents? The World Health Organization defines adolescents as starting from the age of 10 to 19. When I first heard this, I also got a bit shocked because I have a 9-year-old who's about to be a teenager, clearly. This is a phase of life where it's in between childhood and adulthood. So you're no longer a child, but you're not yet an adult. So in the clinical trials that I'm going to speak on, who is considered adolescent is very much protocol dependent. For instance, in the Plus Pills study, the 15-year to 19-year-old were the adolescents that were enrolled in the study. In the Zoli study, it was from the age of 12. The Purpose 1 study, specific focus was focused on the 16 to 17-year-old adults. And it's important to note that the adolescents are not just young adults. They have unique needs that require tailored approaches. So why do we include them in the study in our work that we do? This is a very important time for health development, for health intervention. We live in a young world. It's been estimated that the adolescent population is rapidly growing. They're sitting at about 1.3 billion adolescents globally. In South Africa alone, the population is made up of 10 million adolescents. When we enroll adolescents in studies, we also accelerate the approval of drugs for them. The World Health Organization also recommends that we offer adolescent girls HIV prevention to eliminate the spread of HIV transmission. So I'm now going to move over to speak about the specific studies that I mentioned before. The first up study is the Plus Pill study. Like I mentioned in my introduction, that it was the first oral PrEP study that was on adolescent population in Africa. It provided us with safety information and adherence data for young people in South Africa and the region. It was done in 2 sites. It was done at our site in Masiphumelele and also in Soweto. There was 150 adolescents that were 150 adolescents that were enrolled, and so basically in the first 3 months of the study, all adolescents got PrEP. At month 3 mark, you had an option of either opting out or opting in of PrEP. And what they found is that at the 3 months mark, a few adolescents opted out, about 18% of them opted out. And as time went by, at the 9-month mark, at the 6 months mark, as time went by, what was discovered was that they were more likely to stop PrEP as time went by. And what we found was that mostly is the most factors that influenced it was fatigue. They got tired, they got bored, and also lack of motivation. Okay. So I'm going to quickly go to the next study. And this is one of Catherine Gill's favorite story to tell. The Zoli study, it was a study that was comparing a new drug which for treatment of gonorrhea. This is the first new drug to be developed in over 30 years to treat gonorrhea. And the reason why we need a new drug is because of the emergence of antibiotic resistance to the current standard of treatment that we have, which is the ceftriaxone injection combined with azithromycin. So the study compared the new drug, the Zoli drug, to the standard of care treatment. And why I brought it up is that the study, to be in the study, you had to have gonorrhea. So you had to have symptoms of gonorrhea. So can you imagine if you're a 16-year-old and you have gonorrhea and you have to ask your parent for permission to be in the study? Take informed consent, take it home, and explain to your parent, I have gonorrhea because I'm having unprotected sex with my boyfriend. So because it was a treatment study, we had to involve the ethics committee a lot because there was a standard of care treatment that was provided. We were trying out a drug that we didn't know if it was going to work. But we had frequent progress reports. We made sure that there were protective measures in place to mitigate the risk. For example, there was a test of cure. And also, if treatment failed, we could still treat the adolescents that were enrolled in the study with the standard of care treatment. Okay. So quickly about the drug itself, like I said, is a new class antibiotic that is given as a single dose. So it would help in, according to available data at the moment, there isn't a lot of gonorrhea resistance for ceftriaxone. But even if it was used as a first line treatment, it would actually mitigate the emergence of resistance, of gonorrhea resistance. Okay. So we were one of 5 South African sites out of 15 sites that were enrolling the study in 5 other countries. And out of the 930 participants that were enrolled in the study in all the 5 countries, majority of those participants were from South Africa. But 621 were given the dose of the Zoli, and the other 309 got the standard of care treatment, which is the ceftriaxone injection with azithromycin. So what we found there was that Zoli was not inferior to the standard care of treatment. What that means, it's as good as giving the standard care of treatment. Okay. So quickly about the Purpose 1 study. So we enrolled, so at Massey, overall, Massey contributed mostly to the adolescents that were enrolled in the study. the 16 to 17-year-olds. So as soon as we got a go-ahead from the DMC after the first meeting, we were notified that we could enroll adolescents, which are the 16 to 17-year-old. Like I mentioned before, it is a recommendation by the World Health Organization that the adolescent girls are given HIV prevention packages. So I will quickly going to mention the reason why I'm mentioning the 3P PrEP study was that the site was involved with the McCann Global Health to design a recruitment material that was specifically aimed at adolescents. So in conclusion, we'd like to point out that the key factors when engaging with adolescents are relationships. They need to trust, they need support, they need accessibility, the visit needs to be quick, we can't waste their time, there has to be fun, innovative, and the messages that we put across to them has to be gain-framed for them. So I would like to thank all the study participants for the study that I mentioned, and of course the CRS lead, Catherine Gill, and the site staff, and also the study sponsors. Thank you very much. So the next speaker is Millicent, who is going to be talking about the behavior and biomedicals. Thank you.
Let's see if I can work this. And okay. So first of all, let me say thank you very much for this opportunity. But more importantly, I'm very delighted to see this day, a research day, Desmond Tutu Research Day or Desmond Tutu HIV Foundation Science Day. It's very exciting. So it is really, really a great honor. So my topic is on behavior and biomedicals. This is broad. So what I think I'm going to do, I'm going to talk about how social behavior sciences is integrated in biomedical research, and then I'm going to talk about how social behavior sciences is integrated in biomedicals at Desmond Tutu HIV Foundation and Desmond Tutu HIV Center. And then I'll focus on some of the work that we've done that is linked to biomedical research. I will then talk a little bit about what the future looks like for social behavior sciences. Okay, let me start with a summary of our staffing structure. Usually what we have at the Desmond Tutu, and I say this because we are linked to biomedical research, so I want to kind of pull out those nuances. Usually we have a group of senior social behavior investigators or researchers, and these are sometimes also called project leads. They have specific areas of focus that they focus on within the foundation, and often are linked to different sites that do run biomedical research. We do have a category of PhD students. We have junior social behavior scientists, and there also we have program-specific research assistants. Then at site level, we obviously have sites that are based, staff that are based at different sites. So shout out at the SB, social behavioral scientist at Mercy. Shout out to the social behavioral scientist at Philippi Village, Emma Vundleni, East London, GRO, to mention but a few. I think we do need a strong footing at GSH as well. And usually what happens here is that these operation teams at sites are led by site implementation leads and coordinators, supported by clinicians, research nurses, CNPs, depending on the study that we are working on. And of course, on the other end, you have your research assistants, you have that collect data, analyze data, we have the field team, facilitators, again, depending on a type of study that we are running. Okay, sorry. So I wanted to start by just briefly talking about what social behavior sciences mean. And by the way, let me highlight that when I say social behavior sciences and social behavior research, I use them interchangeably. So sometimes when I say SBS and SBR, they are actually meaning the same thing. But on the broader note, of course, we all know that social behavioral sciences has a range of is an interdisciplinary field. And often what we are looking at is we are looking at how we understand how individuals, groups interact, behave in certain social contexts. But for the purpose of this work, which really focuses on health and biomedical research, I wanted to say that when we are looking at social behavioral sciences in that field, we are looking at the social, cognitive, environmental barriers and drivers that influence health-related outcomes. But for biomedical in particular, which is the topic for the day and the topic that I'm trying to explore, is that social behavioral science is critical because it employs a variety of methods, theories that help us understand. Fantastic, thank you. Yeah, that help us indicate how effective implementation of biomedical research that are essential for biomedical research. Beyond that, I talk about behavior and structural interventions that promote health. And here what I'm trying to indicate, obviously, that is that social behavior research is not just about being connected to biomedical research, but extends beyond that. Okay, so this slide really tries to say that, and to some of the clinicians in this audience, that social behavioral science is not just about qualitative work, right? That we actually utilize a range of disciplines and theories and methods in our inquiry. And some of the fields that obviously we include here, you look at your psychology, sociology, to mention but a few. But in the field of HIV, if you all know, we have obviously worked with a number of theories that help us frame our work, help us understand phenomena on the ground. So you have your individual level theories. I'm not going to mention the details of that, but then particularly in developing countries, we tend to use population level theories. And that's because, of course, we try to understand that behavior is shaped by a number of other factors, not just individual behaviors. Then your methods, the methods that we tend to employ, and we have employed a range of these within the foundation as well, qualitative methods, which is obviously one of the ones that we use. But we also are looking at, we use quantitative methods, use mixed methods, we use randomized controlled trials, not clinical trials, but also interrupted time series. So there is a range of methods that we use. But particularly for this day, which is where I'm talking about social behavior sciences and biomedical research, I wanted to bring a little bit of a historical context to this, right? So obviously social behavior sciences has worked in a number of other areas. So we've worked in cancer. I think in the 1960s, there was great work that was done around the field. But what I would like to say, though, is that social behavior sciences did indeed gain prominence with the HIV epidemic. And these particular papers that I highlight actually speak to that, indicating how social behavior science has been integrated in biomedical research. So our earlier work that integrated social behavior sciences and HIV really tended to center around the knowledge, attitude, and behavior. And I'm not sure whether you remember that. So we had all the CAPS type work that was being done. And what that work led to at that time was the time when we were looking at abstinence, be faithful, condom use. So there was a lot of messaging that was targeting that at the time. But then I think the advances in biomedical research has created this perfect marriage between, not the GNU, but perfect marriage between social behavior science and biomedical research. And of course, this is critical because it was clearly noted that just having medicine and just having pills without people wasn't going to work. It wasn't the magic bullet. The drugs worked, but how are we engaging people? And it was also noting that really diseases are sometimes linked to human behavior. So you really want to look at the human interaction and how that is going to affect biomedical use. I like this quote that I take from this Jade's paper, that when clinicians start treating people, they realize that, oh, actually patients are more complicated than their disease, right? I can treat, it works, but this person is actually more complex, right? So there's need to bring in the social behavior work. Things freeze on me. Let me see if I can use that. Okay. Great. So social behavior research and biomedical research, for those who know the chain of, did I miss something? No. Chain of product development from preclinical to phase 4 being rollout, marketing phase. Social behavioral sciences is in every single step of the product development chain. And some of the studies that we do include descriptive studies, analytical studies, randomized control trials, to mention but a few. But for us to really understand the contribution of social behavioral sciences, given how complex, given how our work could be lost in the clinical world, I wanted us to look at the functional framework by Geist and Stewart. I think these are NIH-based. And I pull out this paper where I pull out this model as well. But what they really try to show is that social behavioral sciences looks at different aspects within biomedical research. So we start at basic social behavioral sciences, and here we are trying to understand populations, priority populations, trying to understand risk. But all that work is actually done to support biomedical research. Then we look at elemental research, where social behavioral sciences here is there for creating interventions that are social behavior linked. So for example, we are looking at interventions like counseling interventions or risk reduction interventions, again geared towards improving biomedical research. And there are many other. We have supporting BSSR, which is aimed at supporting trials as well. And finally, we have integrative social behavioral science. But what I wanted to do on this particular slide is try and show how the work we've done at the foundation and at the center can be traced within those 4 domains of the functional model. So if we look at the basic social behavioral sciences, where we've looked at understanding social behavior dynamics in priority populations, you look at, I want you to look at, so I move from basic social behavioral sciences is connected with how we use our observational and observational studies, descriptive studies, to understand vulnerable populations, to understand how we can engage with community, to understand the language that we should actually be using when we are conducting clinical trials. And some of the earlier work that we did with, we've done under this particular domain, look at work that was done earlier on, I think at Mercy, and I think Karen Middlecoop, Linda Gale-Baker, and earlier people who were at the foundation, looked at HIV vaccine preparedness studies. And here they were looking at how can we engage young people in vaccine studies. We looked at the work that was done at the Youth Center, looking at HIV testing. We look at the work that was done earlier on, the IAVI study that looked at HIV incidence in MSM cohorts. And particularly this also was to try and inform how do we do clinical trials with these populations. Then when we look at elemental social behavior research in the foundation, of course, we've done a lot of discovery and early translation work. One of the preliminary works that we did was the project called Sasha. And here we are trying to look at the acceptability, feasibility of conducting research within adolescents. Here also what you think about, what we looked at was how can we sustain recruitment? How can we sustain retention? What are the ethics of conducting research with young people in this population? We have very many other studies that we've worked on. Then when you look at supporting social behavior research, here this is largely what we've done, and often looked at as though we just conduct qualitative work, but our support has been looking at working with clinical studies like HPTN studies, HVTN studies, MTN studies, and all those we've been able to conduct qualitative work with them. But we also do formative work, right? So the project that was mentioned earlier called 3P, the formative work was largely done by social behavior scientists to understand risk, to understand uptake of oral treatment. Okay, and finally, just to talk about social behavior, integrative social behavior research. Here, this is now where we are looking at rollout, right? Or doing implementation science research. And here, some of the projects that we're working on include like choice and preference of prevention options. We are looking at demand creation. We are looking at real world. How can we facilitate real world implementation? Some of the studies we've done include studies like POWER, I think, your demonstration projects. IMARA has been one of them. We do have Fast Prep, which is an implementation science, but some of the aspects of those projects in that program can actually fit in this particular one. And recently, Affinity, which is looking at long-acting treatment among adolescents. But here, what we are looking at, of course, we look at the feasibility, acceptability in the field. I'm about to finish. All right. So where is the future? We see ourselves, we see social behavior research moving a lot more in what we call biobehavioral studies, right? So looking at behavior interventions that actually do support clinical outcomes or support biomedical research itself. So the intervention is behavioral, but the outcomes are medical. And we already have some studies that we are doing in the foundation, IMARA being one of them, but soon we shall be working on a study called CARES as well. And CARES intends to basically look at supporting choice. So looking at adherence support options that can support the prevention options, HIV prevention options. There is interesting work that is coming around AI. We think we should get involved in that. And then also beginning to look at the fact that some of the models, some of the theories that we have utilized might not be relevant as biomedical research advances. So thoughts around how we can develop new theories in the field. All right. All right, so we trying to create we wanted to share a link of some of the work that we've done that include projects and papers. This link will be shared at some point. So thank you very much.
Thanks, Millicent. Sorry, guys, and I know you have a lot to say, and we're rushing you. So thank you, Millicent. I loved the way you laid that out, and I think with time it's going to be really nice to digest that. So thank you for that. And of course, now we move into the weighty topic of the absent HIV vaccine, but how the quest continues. Over to you, Yashna. No, press the little button.
This
Go
one.
forward.
So, good morning, everybody, and thank you for having me here today and for this opportunity. And thank you for being here and hearing all about our important work. So, there have been more than 250 HIV vaccine trials, most of them early stage, and most of them looking at safety and immunogenicity, so how our bodies or how people's bodies respond to the HIV vaccine. There have been even fewer HIV vaccine trials, about 10 or so, that have actually reached the stage of efficacy. Even fairly recently, we have suffered a number of severe setbacks, but with each iteration of a new promising vaccine candidate, there are important scientific breakthroughs that are being made. So with a host, why am I not getting this? No. Okay, I'll just keep it here. So with a host of new exciting HIV prevention options licensed or in the pipeline, some question whether a long-term vaccine should be prioritized and at what urgency. Our ever-expanding prevention toolkit, which we are all familiar with, now includes injectable HIV regimens. No worries. Okay, thank you. And you will hear more about this from Lulu, and you've heard from Rhandzu already a little bit about the Purpose 1 study. So if everyone who needed PrEP knew about it, had access to it, wanted it, and remained on it for a considerable amount of time, and similarly, if people with HIV persisted on their ART, maybe we would not need an HIV vaccine, but we know that this is not the case. So if we had an HIV vaccine, we would hopefully be able to achieve herd immunity, or in other words, protecting people who were not taking the vaccine. So yes, we still do need an HIV vaccine, and the work still needs to continue. So just to give a bit of a refresher, traditionally a preventative vaccine works to mimic the immune system, or the body's natural response to a virus, which usually results in it being cleared. So what our immune systems do is B cells produce antibodies. CD8 killer T cells would either neutralize the invader or recruit other immune functions to do so. CD4 T helper cells would coordinate this process, and that hopefully results in the infection being cleared. And once that the infection has waned, a subset of B and T cells, memory cells, would stand on guard in case the invader presents itself again. But this doesn't work with HIV. So people with HIV usually do produce antibodies against the virus, but they aren't usually strong enough. So this animation shows the outermost envelope of the protein called the trimer. It should be in gray, and that cycles through different combinations. So the trimer is densely coated in sugar molecules, and that keeps it hidden from the immune system. The yellow and the red parts, which are not coated in sugar, are the parts that rapidly mutate and produce viral mutations. And the antibodies that have been produced by a person usually are produced very slowly, and they aren't recognized by these new mutations. So only around 15% of people with HIV are able to naturally produce broadly neutralizing antibodies. So because there isn't really a model for natural immunity for HIV, researchers don't really have a way to define correlates of protection. In other words, we don't know what HIV protection in a person would look like. Additionally, there are many different subtypes of HIV. Each of them is genetically distinct and occur in different regions of the world. Researchers also have to account for genetic differences in population. So basically, an HIV vaccine that works for one person may not work for the next. So firstly, HIV vaccines won't contain inactivated or whole weakened virus because of the risk for potential for infection. Other ways to target HIV to make a vaccine would use a small part of the virus or using a vector. Nucleic acid vaccines are also an option, virus-like particles, or broadly neutralizing antibodies, which is highlighted in bold for good reason, and I'll get to that later. So back onto types of vaccines, the protein subunit vaccine used part of the HIV envelope, which is necessary for the virus to enter the CD4 cells. Nucleic acid vaccines use small pieces of DNA, that's a DNA vaccine, or genetic instructions for making HIV antigens directly within the cells, that's messenger RNA being used. Viral vector vaccines use a harmless virus or vector which is engineered to deliver HIV antigens directly into cells. Peptide vaccines use short segments of HIV proteins, and virus-like particles mimic the virus's structure, but they don't contain any genetic material. Along with HIV vaccines, there are ways to possibly strengthen the immune system. Prime boost regimens use a strategy that first introduces the antigen to the immune system, and then a series of subsequent vaccinations augment the immune response. Heterologous prime boost regimens combine different types of vaccines, and mosaic vaccines incorporate genetic material from multiple HIV strains. So looking at vaccine development timelines since the late 80s, where HIV was discovered to be the cause of AIDS. The first HIV vaccine candidates were the protein subunit vaccines, which target the envelope, which is necessary for entry into the cell. And the first African HIV prevention trial was conducted in Uganda in 1999. And around the same time, we had the HIV vaccine trials network being established, along with smaller vaccine groups in Kenya and South Africa. The Thai trial, infamous Thai trial, RV144, gave us the first positive results from an HIV vaccine, efficacy results in the early 2000s. And fast forwarding to 2010, we had the discovery of VRC01 and VRC02, broadly neutralizing antibodies in people with HIV. So the Desmond Tutu HIV or Health Foundation has been involved in HIV vaccine research since 2006 with the first IRV trial. Also fast forwarding again to 2015, where the HVTN 100 trial looked at the safety and immunogenicity of the LVAC canarypox viral vector vaccine, plus a protein subunit vaccine. So this was a heterologous prime boost regimen. And this was the same regimen used in the Thai trial. But this was adapted to also target subtype C, which is the predominant subtype circulating in sub-Saharan Africa. So for this trial, the immune response criteria were met, which meant that this led to the development of HVTN 702. 702, or Uhambo, started in 2016 at our Emma Vundleni research site. It used the same regimen, but investigated the vaccine's efficacy and safety to prevent HIV in people who found themselves in situations that gave them high levels of acquiring HIV, high risk. Sadly, the trial was stopped early because of poor efficacy. So rates of HIV infection in the placebo arm were similar to that of the vaccine group. Along came Imbokodo, HVTN 705. This was also a phase 3 safety and efficacy trial conducted in young women having sex with cisgender men. An adenovirus vector vaccine delivered a mosaic of HIV antigens from multiple strains, trying to cover a lot of HIV diversity. Unfortunately, the trial ended prematurely because of poor efficacy. And just a little break into the discovery of broadly neutralizing antibodies. So monoclonal antibodies are derived from a single cell line, which gives them defined specificity. And since 2010, researchers have isolated, sequenced, and synthetically made VRC01 and VRC02. These BNAbs have been administered by way of passive immunization, so bypassing the active immunization, which relies on the immune system to recognize and make broadly neutralizing antibodies itself. And currently, multiple doses of BNAb seem to be required to maintain immunity in people who receive them. But these studies have provided valuable feedback and guidance as to what the next immunogens in the sequence may look like. Okay, back to our trials. So the AMP studies, or antibody-mediated prevention studies, HVTN 703 or HPTN HIV Prevention Trials Network 081. These were proof of concept trials, and the Grooterskeer Clinical Research Site participated in this trial. And this one looked at safety and efficacy in young women having sex with cisgender men. And this was one of the first studies that I worked at DTHF. VRC01 was overall not found to be effective in preventing HIV, but it did reduce the risk of acquiring HIV in a small subset of strains classified as highly sensitive to VRC01. So building on from the AMP trials, the field continues to isolate highly potent BNAbs, which are able to provide sterilizing immunity at low doses in non-human primate studies. Researchers have found that by combining antibodies, we can make them more stable, more potent, more durable, and offer various formulations, for example, subcutaneous injections, which can lower the cost of manufacture and increase tissue penetration. So there are new approaches to BNAB design, and one of them is germline targeting. So basically what happens is a primer vaccine recruits naive B cells, and the B cells go off to the lymph nodes to mature, and then you have a series of booster regimens or vaccinations that train the B cells to recognize the right antigens and to produce BNAbs from that. So the WHO have come up with a document, the preferred product characteristics for monoclonal antibodies for HIV prevention, preparing for when we have a safe and effective vaccine. In summary, BNAb should be used for the prevention of HIV in adults and in exposed neonates and infants, and be inclusive of priority populations, including adolescent girls and young women, and gender diverse groups. It should also be cost effective to manufacture, thereby decreasing costs in low to middle income countries. They should cover a broad range of strains of HIV globally, and preferably a single injection given at a single site at a single time is preferred. And because giving IV formulations because intravenous formulations require more expertise and more supplies, subcutaneous and intramuscular injections are preferred in LMICs. So this is all well and good, but translating this into equitable access requires more advocacy, more partnerships, and the strengthening of our health systems. Most biomedical manufacturing occurs in the global north, but there are initiatives to strengthen this capacity in Africa. In fact, the brilliant consortium of the South African Medical Research Council was established last year, and they are predominantly female-led group of African scientists whose goal is to design and test HIV vaccine candidates from Africa. So we are fortunate enough to be one of two sites in the 8 African countries. We are one of 2 sites in South Africa, which involves 8 African countries participating in their first project. So with that uplifting news, I will leave you with the wise words of our patron, the late Archbishop Desmond Tutu. Hope is being able to see that there is light despite all of the darkness and all of our vaccine failures. Thank you. Okay.
Thank you. That's known as the glass half full as opposed to the glass half empty. So thank you for that, Yashna. Yes, and many people may be saying, well, you know, is there still room for vaccine discovery? And I'd be happy to have that discussion with you at lunchtime if indeed you want to debate that one. Right, well, we're moving right along. So from the vaccines into the other biomedical prevention, and hand over to Lulu.
I think it's afternoon now. Good afternoon, everybody. So I am going to be speaking about...
Press again.
I'm going to be speaking about the prevention revolution. So before I can speak about the prevention revolution, I think I need to talk about the evolution of prevention. So the beginning of my talk will focus on that, and then I will move on to the PrEP studies that we've actually undertaken as an organization. So the first 30 years of the HIV epidemic focused on treatment, and the goal of ending the HIV epidemic is becoming more attainable through decreased transmission and HIV-related deaths. Great gains have been made, with life expectancy having increased markedly and having almost doubled. But in spite of this, 1.3 million people became newly infected with HIV in 2023. And globally, 44% of all new infections were seen among women and girls. And young women and girls in sub-Saharan Africa remain disproportionately impacted by HIV when you compare them to their male counterparts and women in the rest of the world. So now moving on to the historical development of vaginal microbicides. As a result of this disproportionate impact on young women and girls in sub-Saharan Africa, early HIV prevention biomedical strategies focused on female-initiated and controlled methods, namely microbicides, which would be safe, which would be effective, affordable, easy to manufacture, stable under different environmental conditions, and of course, comfortable to use. However, many microbicide options failed due to the lack of their protective effect and unacceptable formulations. So if we look at the slide, the first three candidates for microbicides actually were ineffective. If we look at the surfactant nanoxyl 9, it was actually shown to cause some disruption of the vaginal epithelium and was harmful. The other products, those that maintain the pH in the vagina and so on, like buffer gel, all of those were ineffective in protecting against HIV, and so the development of those products was stopped. Our first real success was seen with ARV-based microbicides, like entry inhibitors like Maraviroc, MVC, tenofovir, and the dapivirine vaginal ring. Okay, in terms of the pipeline, what we have a fantastic toolbox of currently available products, which includes treatment for prevention, PEP, daily oral PrEP, and what we have coming in the future, recognizing that choice is important for adherence and persistence, we have a whole toolbox of stuff that's coming in the future, and that includes long-acting implants, multi-purpose and long-acting vaginal rings, and broadly neutralizing antibodies, as Yashna has already mentioned. So what we do have now, and which I'm going to be talking about in detail because we've participated in those clinical trials, is the dapivirine vaginal ring and the long-acting injectable cabotegravir. What's not shown on the slide, but it's not yet registered, is lenacapavir, which should be included in the slide. Okay, so before I move on to those clinical trials we've participated in, I think it's important to talk about choice. And we've learned a lot of lessons from contraceptive uptake. We have an IUD, implant, oral contraceptives, and so on. And what we've seen from that is that from providing contraceptives is that choice is important for increased persistence on a chosen method, and with every method that is available, there is a 12% increase in use. So similar to contraceptive needs, people require methods or formulations for HIV prevention which change at different points in their lives. So I'm going to talk about oral PrEP, and this was the first biomedical technology that was approved by the US, by WHO and FDA between the periods of 2012 and 2015. So this is oral PrEP contains Truvada contains two different ARVs from the same class of ARVs, the NRTIs. And the dosing strategy is that it's a daily oral pill needs to be taken. So multiple trials have looked at oral PrEP in combination with other products. So like Voice looked at oral PrEP Truvada, comparing it to tenofovir gel. And so and in all of these studies that looked at oral PrEP, what they found was that with increasing adherence to oral PrEP, the efficacy or how well it worked increased as well, so that efficacy could be as high as 90%. Okay. So For people assigned male at birth and not on gender-affirming hormones, event-driven or on-demand PrEP is recommended. And this recommendation is based on a clinical trial, a French clinical trial, which was done among MSM, and it showed an 86% relative reduction in HIV in comparison to placebo. So the regimen involves taking 2 tablets 2 to 24 hours before sex, 1 tablet during exposure, and 1 tablet 2 days after exposure. So I wanted to mention Descovy because, or FTAF, which I'm going to mention when I talk about the Purpose 1 study. FTAF is a combination of 2 antiretroviral agents which belong to the class of NRTIs. Just like Truvada, it contains emtricitabine, and the other NRTI is tenofovir alafenamide. That's where we get the TAF from. So it can also be used for daily PrEP or on-demand oral prevention. The advantage of FTAF is that it has better PK and fewer side effects in comparison to oral Truvada. The recommendation for FTAF was based on a study done amongst MSM and a transgender woman. It was called the Discover study, and it enrolled over 4,500 individuals, and this study showed that FTAF or Descovy was non-inferior to Truvada in preventing HIV infection. So FTAF or Descovy is licensed for HIV prevention in men, but not yet licensed for use in women, because there have been no successful clinical trials showing that women can adhere to it and that it would prevent HIV infection. So I'm going to move on to a study that we've actually undertaken around the dapivirine vaginal ring. So in the first quarter of 2021, the dapivirine vaginal ring was approved for HIV prevention by WHO for women at substantial risk of HIV infection. And this was based on 2 clinical trials which we were involved in. These studies, the Aspire study and the Ring study, were run concurrently, and they enrolled over 4,500 women. Women were randomized in a 1:1 ratio, either to the ring or placebo. So both of these studies showed on average a 30% reduction in HIV risk, but subsequent analyses showed a higher efficacy with consistent use of the ring over 28 days. So the relevance of exposure to different PrEP methods and the desire to be able to, or the ability to be able to choose a method was demonstrated in an adolescent and young women study of the vaginal ring and oral PrEP. In this crossover design study called REACH, women were randomized to either Truvada or oral PrEP, to Truvada or the ring for a period of 6 months, and then they were allowed to switch over and use the other product for another 6 months. And then finally, in the last 6 months of the study, they had the opportunity to actually choose whether they wanted to use the ring, take oral Truvada, or not use anything at all. And so given this choice, in the study, twice as many adolescents chose the ring over oral PrEP. So I think I need to speed up a little bit. And the second pivotal study that we did was involved CAB-LA. So this is a strand transfer integrase inhibitor. It's long-acting, and the dosing strategy is a single injection is given every 8 weeks. And the results of 2 pivotal studies, one HPTN 084, which enrolled women, and then HPTN 083, which enrolled MSM and transgender women showed that CAB-LA was superior when you compared it to Truvada. Okay. Then moving on to lenacapavir, as Rhandzu mentioned, we've undertaken the study at three of DTHF sites. So it's a long-acting 6-monthly injectable for HIV prevention. It's a novel drug in that it acts at several points in the life cycle of HIV. It's given subcutaneously. In the research studies, it was given by research staff, but there is the possibility that individuals could be trained on how to administer self-injections. So the thing about this drug is it is, apart from its novel mechanism of action, is that it has a high resistance barrier. And, but we need to bear in mind it's a single agent for HIV in prevention when you compare it to the other strategies which have multiple combinations of multiple ARVs. So very exciting to have been involved in this study, and for the first time we saw in the study among women that an HIV biomedical technology offered 100% protection against HIV. In the study done amongst MSM and transgender women, it showed a 90% effectiveness in preventing HIV infection. So I just wanted to talk about adherence because, especially to the oral product in that study. So in Purpose 1, amongst women, there were actually 3 study products. So it was the injectable lenacapavir, it was Truvada, and it was Descovy or FTAF that I mentioned before, which is only currently licensed for use in men. So the study, people were randomized in a 2:2:1 ratio, lenacapavir, Descovy and Truvada in that study. And what we found was that adherence to injections was high, and how we measured that was by how many people returned to get their injection every 6 months over a period of 1 year. And the timeliness of injections was over 90%, so people were very adherent in the study. But what we did find that was that adherence to oral study product was very poor, in that you see that for FTAF and both for Truvada, so in the black bar, that's low adherence, where people took just less than 2 doses per week. And then in the yellow bar there, it shows medium adherence with greater than or equal to 4 doses per week. And then 2 to 3 doses is medium adherence, and high adherence in the light blue or turquoise is greater than 4 doses per week. So what we did see for oral study product was that adherence was poor in general, and as time went along, this adherence got worse. Okay, so coming back to what is the future of FTAF for women, and so they did a small sub-analysis in the study where they looked at the 37 people who seroconverted, who had received FTAF and seroconverted. They looked at adherence in that group and compared it to matched controls of 159 individuals who had received FTAF but had not seroconverted. And here we can see that, Okay, so here we can see that adherence to FTAF was in the group that seroconverted was very poor, and those who did not seroconvert had better adherence to FTAF. Okay, so we, as an organization, were involved in all of these pivotal studies, all of the Depoverin ring studies, the CAB-LA studies, as well as Purpose 1 and Purpose 2. As an institution, we contributed to the development of the primary manuscripts in addition to secondary manuscripts. We also disseminated results to the scientific community, stakeholders, and general communities through conference presentations and other meetings. So finally, there are several options in terms of PrEP that are coming in the future. And PrEP is only one part of HIV prevention. To facilitate PrEP implementation at a program level to increase adherence and persistence, WHO has recommended that services be demedicalized, decentralized, meaning that access should be increased. And I think Elzette is going to talk about that next. So in summary, why are we calling this a PrEP revolution? We are calling it a revolution because since oral PrEP was approved in 2015, in a short space of 2 years, we had 2 long-acting products being made available, the Depivirine vaginal ring and Cab-LA, and now we have lenacapavir, which would soon be registered for use. In addition, we have choice. We also have tailored approaches addressing intermittent exposure risk, such as single or event-driven PrEP is now available to men. And in addition, we're not following the old-fashioned model of doctors and nurses providing PrEP. We're moving to a decentralized, differentiated service delivery model to increase uptake and persistence. On behalf of the people I work with who have so much of passion for this, for HIV prevention work, I'd like to say thank you for being here today and listening to the work that we are undertaking. Thank you.
Thank you. Afternoon, everyone. So I'll be speaking about taking it to scale and beyond. So as Lulu mentioned, so in South Africa at the moment, we have these 3 PrEP products approved. The oral PrEP, we have the Depivirine vaginal ring, and then we have injectable cabotegravir. So Desmond Tutu Foundation is also an implementing partner for the National Department of Health, which means we go into the real world and try to understand then what happens when you put these products into the real world. So I was asked to speak about scaling up, but then I changed it to also speak about scaling out and scaling deep. Wrong way. Okay. So these are brilliant products, very, very exciting, 100% no infections, but it doesn't help if we do not get them to the people that we need to reach. And I really like this graphic that speaks about not hard to reach groups, but really difficult to reach some services that people need. And if we want to reach very ambitious targets for HIV prevention for 2030, we will need to adjust how we engage with people, and this includes well-designed, well-targeted, people-centered services. So as Lulu started mentioning, we then start looking at what does the product look like? Can we have longer product? Where do we deliver it? Who delivers the product? And the what of it. So just to explain, we have scaling up, which we know. Now we have a product and we want it everywhere. Right? So we know this product works, there's evidence that it works. But scaling out then means you use that same product that you have evidence of and you scale it out into different settings. So not just in the public health setting, but you go look at, does people want this at their homes? Do they want it from a mobile clinic? So that's scaling out. And then I also just want to reflect on scaling deep, which is we need to understand the person that we are scaling this product to. So now if we want to scale deep, we start getting into who are segments of people that we want to reach with a product and how best do we market and get those products to different people. So getting into understanding your audience, so the people that the product will benefit, who are in need of this product. So we all have seen different variations of speaking about the structural constraints when a person wants a product. So we've put a lot of saying person has individual choice and agency, but we need to reflect on all the structural community, school, norms, etc., that might be standing in the way of this person reaching the product that they need. So some of the work that we have done when we've provided, in this case to young women, a variety of settings where they could get oral PrEP in this case. It included a mobile clinic, courier service, one government facility, and a youth club. We started looking at who are the different people that come back and where do they go for their services when they can move around. And now we start getting into scaling deep. Who are these people? How do we market it? And we found some convenient PrEP users. They just wanted the easiest way possible. Your social PrEP user wanting to do this with their peers or friends or wanting a chat with the provider or the counselor. Then there's the independent PrEP user. Please send this to my home. Don't talk to me a lot. I know what I'm doing. I just need the product. And then the discreet PrEP user, where this is really someone who is not disclosing that they're using PrEP, and we know how important that is for social support. We frequently see that they have controlling sex partners, so really a group that we need to get to. Now, still scaling deep is we need to understand, again, who's the audience. So before we started our current large fast PrEP project, we put together a youth reference group. So they were there before we decided anything about how this project will look. We just knew that we were going to have this product. So this was back in 2020 and 2021. And we still have them going. It wasn't just a tokenistic ticking off the box that we did. We still have them going. They are the youth experts. We consult them at least twice a month with all demand generation, with all services that we do. They check on our research. They do the member checking on that. And sometimes they also act as secret shoppers, visiting some of our services and reporting back with feedback. Was it adolescent friendly for them when they came to our services? Then there's demand generation. So now maybe we know a bit about the population, but how do we let them know that we know about you and we have something that you want and need? So in our demand generation, we are really directing the target population then to our services. We tell them about where we will be in the week. There's a weekly schedule. We send out empowering messages. We send out messages about how are you adherent, how do you remember to come for a refill. We put out, when there's a new product, especially like Behaviour or Cabernet, we put out awareness of what is this product, how will you start deciding what product you would want. So now I'm going to get to the scaling up and scaling out in one. So Fast PrEP started in 2022. And here the aim is to really evaluate if we work in one district, which in this case is the Klipfontein Mitchells Plain district, and we offer a choice to young people of different places where you can get your PrEP to understand will that lead to better persistence, because now they can choose a place they would want to be. And then in time also rolling out newer PrEP modalities like the vaginal ring and the injectable PrEP. So our current rollout, we started in 2022. So we have 4 mobile clinics, we have youth clubs, we are in 16 schools, at 12 government health facilities, we have a courier service and 2 quick depots. And here we are targeting a general population between the ages of 15 to 29 years, allowing a little bit of an older age for men, as we know that they are the partners of the younger women. So to date, just on the mobile clinics, we've provided to sex reproductive health services to almost 17,000 young people. And we've had close to 14,000 people initiating PrEP from the services. And that is, as I said, we are in different locations. So the uptake has been either from the mobile clinics, the majority of the uptake has been there, and then 3,000 in the public health facilities. So in those facilities, it's government staff. We've just placed 2 peer navigators in each of those facilities to assist young people. So I'm just going to, this is early days, we're still busy with this project, so just sharing a bit of information of what we have found so far. So we started looking at who are the people that come to the different services at mobile clinics compared to government health clinics. We've seen that majority of adolescent young people, as I've said, go to the mobile clinics in their community. We also see more men go to mobile clinics. We got more people who have casual sex partners. People with STI symptoms are more likely to go to the mobile clinics. People who show high rates of alcohol abuse, and also people with moderate depression and anxiety. Then people with moderate depression and anxiety and pregnant women were less likely to go to the mobile clinics and more likely to go to the facilities. So getting into this information, we understand the wraparound services, again, that some people want. It's not just about getting the HIV prevention, but we see STIs here, we see needed interventions for alcohol use, we see needed interventions for depression and anxiety. Then as we started this work, this is some work from my colleague K2 up there. So we started looking at, as you increase access now, what will your continuation on PrEP look like? So we divided the work into 4 cohorts. We started off initially where we were just in a few government clinics and then 2 mobile clinics, and then expanded our services. And what you can see in the Kaplan-Meier is, as we increased the number of places that young people can get services, we did start seeing better continuation on PrEP. So really speaking about the scaling up, yes, but also scaling out into different spaces and making it convenient and easy to get their PrEP. And then what is next? I was just speaking about oral PrEP at the moment. In September of last year, we also received a donation of the Depivirine vaginal ring. So we've started rolling that out on mobile clinics and so far in 2 government health facilities. The partners here, we are very happy to put it in more facilities if you want. And then also the injectable cabotegravir, we have rolled out into 1 mobile clinic and 1 government health facility. So using that same model of making it easy for people to get to the PrEP that they need, as well as also looking at who are the people that go where, who are the people that's choosing which products, and how do we best, again, market it for them when we get to wider scale up of these newer products. I just want to acknowledge everyone in this project. It's a huge team. We are more than 70 people in the Fast PrEP team, but also a special shout out to the mobile teams and their leaders, the peer navigators in all the facilities. There's 24 peer navigators in government health facilities and in the youth reference group, and our Western Cape Department of Health stakeholders and partners. Thank you.